In today's lecture, we are going to know what is a lambda phage. Lambda phage is an enterobacteria phage lambda, officially known as Escherichia virus lambda. It is a bacterial virus or a bacteriophage that affects the bacterial species, particularly E. coli. It was discovered by Esther Lederberg in 1950. The wild type of this virus has temperate life cycle that allows it to remain either inside the host or outside the host. Let me put it in straight words. There are two phases for this virus or for any virus as a matter of fact. The two phases are lysogeny and lytic phase. In a lysogenic phase, what happens is a virion enters into the host and completes all the steps of the replication, followed by its stay inside the host. That means it does not leave the body of a host. Contrary to it, there is lytic phase, in which a virion enters into the body of a host or the cell of a host. It completes all the cell cycles like as attachment, penetration, uncoating, replication, assembly, and then exit. The exit is the main part present here because due to the presence of exit phase, the lytic phase gets complete. That means there is a difference. In lysogenic phase, the virion does not leave the body. In a lytic phase, the virion does leave the body. The phage has a structure in which there is a presence of a head known as a capsid, a tail and tail fibers. The head consists of double-stranded linear DNA genome. During infection, the phage particles recognizes and binds to its host. How will it bind to the host? By joining itself with the receptors present on the surface of the host. Usually, a lytic phase ensures where the lambda DNA is replicated and new phage particles are produced within the cell. This is followed by cell lysis. What happens is that a virion cannot enter the cell or cannot leave the cell without causing the lysis of the cell. However, under certain conditions, the phage DNA may integrate itself into the host cell chromosome, that is a lysogenic pathway. In this manner, the lambda DNA that is in a lysogenic pathway is called a prophage, and it stays inside the host genome without causing apparently any harm to the host. The host which contains a prophage is known as a lysogen. A lysogen can only enter into the lytic cycle under the stressed conditions. What is the anatomy? The bacteriophage lambda consists of proteins and various copies. The virus particle consists of a head and a tail that has tail fibers. The whole particle consists of 12 to 14 different proteins with more than 1000 protein molecules present inside a DNA molecule. The layout of lambda phage is linear, consisting of operons, promoter regions, and capsid coating genes. It consists of 48,490 base pairs, having double-stranded linear DNA with 12 base single-strand segments at both 5' ends. How does this cause the infection? Or we can say, what is the life cycle? The lambda phage or the J protein present in the lambda phage interacts with lambiporin. Lambda phage is a non-contractile tailed phage, means during an infection it cannot force its DNA through a bacterial cell membrane. If there is already an existing pathway present, that way only the virion can invade the host cell. Otherwise, it cannot invade the host cell. Bacteriophage lambda binds to E. coli because by the means of J proteins present in the tail tip, the J protein can insert itself into the host. The J protein interacts with the maltose outer membrane porin, that is a molecule which is a part of maltose operon. The linear phage genome is injected through the outer membrane. The DNA passes through the mannose permease complex in the inner membrane and immediately circularizes. Lambda phase DNA injection into the cell membrane using mannose PT permease, that is a sugar transporting system, which means it's a mechanism of entry into the cytoplasm. After this, the transcription starts, but there's a presence of N protein, which is a primary factor for transcription. The N protein is an anti-terminator and it functions by engaging the transcribing RNA polymerase at specific sites. The C3 protein acts to protect the C2 protein from proteolysis by FTSH. What is FTSH? It is a membrane-bound essential E. coli protease, which functions as an inhibitor. The stability of C2 determines the life cycle of a phage. What is N anti-termination? N anti-termination requires the assembly of large ribonucleoprotein complex that can effectively prolong the anti-termination process. How does it occur and why does it occur? This occurs without the N protein interacting with the DNA. As we read earlier, we need an N protein to interact with the DNA. The protein binds to the freshly transcribed mRNA. N protein binds to box B in each transcript and contacts the transcribing RNA polymerase via a RNA looping. The entire complex continues transcription, but it can skip through termination sequences. What is a lytic life cycle? As we read earlier, there are two life cycles, that is a lysogenic life cycle and lytic life cycle. In a lytic life cycle, the phage follows most infections where C2 primer protein does not reach a high enough concentration due to degradation, so it does not activate its promoters. This means that all the steps are followed and the exit is a primary coefficient. The ONP proteins initiate replication. Transcription from pre-R promoter can now extend to produce mRNA for the lysis and the head and tail proteins. There are many other products or genes present inside it, whereas S, R, RZ and RZ1, which cause cell lysis. We have read earlier that cell lysis is a very important thing for this whole process to occur. Other than this, there are various other questions you need to answer. First is describe what is a lytic phase, explain lysogenic phase, describe the structure of lambda phage, what is a lambda phage, and explain the life cycle.